Hello guys, this is Sonu, and today I'm going to discuss about very important topic in the pharmacology, that is the antimicrobial drug. As we know that the microbial diseases is a very common diseases in the all over the world. Even in the small changes can be attack the microbes and produce the diseases. As we know that our human bodies a lot of microbes are present, but it can try to penetrate over the cell and start to produce the diseases, but our defense mechanism and our antibodies protect from them. But due to some changes, for example, if any person is suffering from immunodeficiency, on the time whatever the microbes are present over the body, they can easily penetrate over the cells over the body and start the producing the disease. So on the time we have to take the antimicrobial drug, or maybe from the external environment, maybe due to the lot of pollutions or maybe some weather changes. On the time they can enter over the body and start the different types of diseases. So on the time also we have to take antimicrobial drug for the protection or for the stop the diseases. That's why it's very important to know about the antimicrobial drug. So let's talk about that. What is the antimicrobial drug? It is an agent, we can say, that the kill the microorganism, or we can say it's inhibit the synthesis of the microbes. It can be synthetic, it can be semi-synthetic, or it can be natural also, natural source. So this is the antimicrobial. And antimicrobial drug means, we can say, it can be different types of microbes can be killed. So let's talk about that which types of microbes can be killed. So first of all, that bacteria, because it's a very common. So bacteria. The next one, virus. Then fungus. Then can be protozoa and helminthes, or we can also say parasite, sorry. So bacteria, virus, fungus, protozoa and helminthes, the all microbes, if we kill, if by the drug, if the kill the any five of them, we can say it the drug is the antimicrobial drug. Means if the kill the bacteria, so we can say antibacterial, virus antiviral, fungus antifungal, the the protozoa antiprotozoa, and helminthes anti-helminthes or anti-parasitic disease. So the all of the antimicrobial drug, so we can say they all of belong to the antimicrobial drug. But in some time they confuse with the antibacterial, antibiotic. We can say that all antibiotic is belong to the antimicrobial drug, but we cannot say that all antimicrobial drug is the antibiotic drug. It's totally wrong, absolutely wrong, because all antimicrobial drug is the, it can be antibacterial, antiviral, antifungus, antiprotozoal and antiparasitic or anti-helminthes, but not particular we can say antibiotic. But all antibiotic drug, we can say that this drug is belong to the antimicrobial, but all antimicrobial is not belong to the antibiotic. This is the very important information and very, we can say, confusion. They always we confuse with the antibiotic and antimicrobial. That's why we should know. So now, and firstly we'll discuss about the antibiotic, because antibiotic is the more common diseases. Bacteria is the more common diseases than the other virus, fungus and protozoa and helminthes. So first we'll discuss about the bacteria, then we'll further move other microorganism. So before start the antibiotics means drug name, first we should know that what is the criteria, what is the main characteristics of the antimicrobial therapy. So let's talk about first of all them. Okay. So the main characteristics of the antimicrobial diseases, that is shortcut antimicrobial disease. First, it should be selective toxic to the microbes, and it should not be, it means it's non-toxic for the host cell. What is the host cell? Host cell our the human cell, our the human healthy cell. So whatever drug, whatever the antimicrobial drug, first it should be the attack mainly the microbes. It's a more toxic for the microbes and the less toxic or non-toxic for the host cell. The first criteria. Second one, it should be microbicidal than the static. What is the microbicidal and microstatic? So cidal means cidal. Cidal means the kill the microbes, and the microbiostasis means, so a stasis means inhibit. If we compare with between two them, obviously kills the better because we're killing the bacteria. But a stasis means the just inhibiting the, we can say, the growth of the bacteria. So let's talk about, for example, that this is the skin, and here is the any abscess of, let's talk about, here is the any wound is there, and in the wound a lot of bacteria is there. This is the all the bacteria. So if we're giving the microbial stasis drug, means they just means in this wound a lot of bacteria are producing and accumulating of the bacteria. Concentration is the very high of the bacteria. So if we will give the microbial stasis drug, so on the time it just inhibit the proliferation, or we can say inhibit the synthesis of the new bacteria. Means whenever the drug the efficacy of the drug and action of the drug, maybe for example, if you give the 5 tablet of the microbial stasis drug, means they're just inhibiting the bacteria. So after the 5 day, for example, the drug is the taking the person is the one tablet in 1 day. So after the 5 day, whenever the drug action is become very low, on the time again the bacteria will start to growing. So it will not good for the person suffering from, means the wound heal become very slow, because just we inhibiting the bacteria proliferating, or the replication of the bacteria, synthesis of the bacteria. But in this conditions, if we give the microbicidal drug, means they will kill the bacteria, so it's a much better, because in this area if they kill the bacteria, so heal of the wound is very fast recover wound heal. That's why it should be bactericidal than the the microbicidal than the microbiostasis. This term the same we can apply in the bacteria, because in the bactericidal and bacteriostasis we'll discuss later when we will discuss the bacteria. Next one, it's a properly delivered to the site of infections. Means the drug should be very easy to penetrate over the cell and where the infections is there, because then it's a very fast recover will be occur. So for the proper deliver, it should be good soluble in the blood and good soluble in the body, it any cells to penetrate. How it's for what we have to need for the good soluble, as we know that drug should be lipid the lipid solubility, and drug should be unionized, means it should be non-polar. If it will be polar, so it will be ionized, and as we know that the polar compound is a charged compound. So for the charged compound penetration we need active transport, we need energy. So on the time it's not good penetration. So for the good penetration, drug should be lipid solubility and unionized. These two things are very important. Because why the lipid? Because our normal cell also made by the lipid, and even bacteria and some all microbes are the cell is the made by the lipid. That's why it's very easy to penetrate and it can be kill the bacteria. And the same thing unionized, because non-polar, so without any active transport, by the passive diffusion it can be enter the any cells. Then it should be not cause allergy and other effects, obviously, because a lot of drug can be start allergy, that is the hypersensitivity. So if the allergy will be developed, so obviously it will not good for the person or for the patient. And other effect, for example, a lot of drug are inhibit our normal flora in the GIT, and produce different types of effect, like the diarrhea and some vomiting, and some other infections can be created. That's why it also should be avoid. Then it should be without resistance. Obviously if drug will be resistance, so it will not helpful for the person who suffering from microbial disease. Even it will become too worse, because in one side they're suffering from microbial diseases, another side drug become resistance, and if you're giving that person, so drug will be just accumulate in the body and start to the different types of side effects. So that's why it become too worse. That's why drugs should be without resistance. Then it should be not develop super infection. What is the super infection? As I said, that other effects, super infections. So let's talk about what is the super infections. Super infections means we can say that in the GIT, this is the GIT, and here is the normal flora. Normal flora, and in the normal flora, the function of the normal flora, here is they're producing a lot of protein, they're producing a lot of vitamins. So if by the drug, if we inhibit the normal flora, the functions, so what can be happen in this condition? And even a lot of bacteria is there, but they will not harmful because they're protecting the normal flora. They are a lot of releasing the protein, so by them they're protecting. But if we will inhibit the normal flora by the drug, because it can be most probably if you increase the amount of the dose, or maybe a lot of combination, 2 and 3 combination of the drug, if you're giving the person on the time, this problem can be occur. So in this condition, if you inhibit, so a lot of whatever the function of the normal flora, it will be obviously inhibited. And in these conditions, some of the fungal, some of the bacteria are attacked in this area because they are not working well and start the super infections. So super infection, most probably the fungal disease can be appear. For example, candida, candiditis, candida. You can remember from this is the fungus. So in these conditions, super infections can be developed and it can become too worse. That's why it should not be developed super infections. And this types of problem most probably occur whenever we use antibacterial, most probably. So these are the main characteristics of the antimicrobial treatment or antimicrobial therapy. So out of this one, if anyone will be not we can say apply, means for example, if not selective toxic for the microbe, so it will just create the side effects. And another thing, if the microbicidal is not, and if the microstatus is the more, so as I said that it's not well, because it can again start to producing synthesizing the bacteria or synthesizing the microbes, and the condition become again will start. Then obviously if drug will not lipid soluble and not unionized, so it will not good for the person. For example, any person suffering from meningitis, so in these conditions, we have to give those drug who is penetrate the blood-brain barrier, because meningitis suffering from the in the brain. So by the CSF, cerebrospinal fluid, they will go and they kill the bacteria. So in these conditions, if drug will water soluble, means a hydrophilic drug, or maybe it will ionized drug, means the charged drug. So in this condition, it will not very poor penetrate in the blood-brain barrier, and they will not treat the. I mean, it will not affect for the meningitis. Then should be caused if the person will cause any allergy. So for example, if you're giving the drug, any out of the any antimicrobial drug, and the person start to allergy, so on these conditions, they will not, obviously allergy is the very dangerous diseases, we can say. If the type 1 hypersensitive can be occur like anaphylactic shock, so sometimes patient can be die also. So in these conditions, we have to select, or we have to first test the drug. For example, if you're giving the person, like the first person, if any person to come our clinic, and we don't know about the which types of drug is the allergy for the that patient, so in the time first we have to give very short dose. For example, the normal dose is the 100 mg. So first we either 10 mg or 20 mg or maybe 30 mg we have to give, and just we have to wait. If everything is okay, then we can start the normal dose. So by this way we can say see the person that the person have any allergy of this drug or not. If because we have a lot of antimicrobial drug, so we can change also. So for example, if any antibiotic, because antibiotic have a lot of classification, a lot of drug which belong to the antibiotic. So one antibiotic, if giving, for example, beta lactam, that is the penicillin. So if we're giving the penicillin and the person say that we see that the person have allergy from the penicillin, so on the time we can just change the another group like azolad, macrolad, and some other tetracycline, and some chlorpheniramine and fluoroquinolone, lot of drug. So but like that we can change, and we can just try to prevent the allergy. Should be without resistance. Obviously we have to use like that the person will not develop any resistance. How the resistance develop? It's most probably due to the lot of combination of the drug, or due to, for example, as we know that the person have this bacteria, and if you giving those drug is not good, or we can say not the killing that bacteria, it's not well killing of for the particular bacteria. So like for example, let's talk about that a lot of mechanism of the developing of the resistance, but I'm just talking about that simply method that how it can be developed. For example, the person have a staphylococcus, and if we have two types of antibiotic, one is here and B is here, and as we know that if we will give the B antibiotic, it will not well kill the staphylococcal. It will not good kill the staphylococcal. And as we know that if we will give A antibiotic, type 1 A means A antibiotic, A group of the antibiotic, it will easy to kill the staphylococcal. So better to give this one. If you will give this one, maybe it can be develop resistance. That is the first mechanism. Some other mechanism also, because bacteria can change on the our body structure, on our we can say the cell wall. On the time also it can be, develop the resistance. We will discuss about each bacteria or each drug, that which types of drug is most probably develop the resistance in which bacteria or which microbes. Then next one is the should not be developed super infections. Obviously, as I said that in the most probably the antibiotic cases they develop super infections. So these are the major and main and very, we can say, important criteria that we have to maintain this types of characteristics. Now let's talk about that. About the first, we'll discuss about the bacteria, means the antibiotic. Then we'll further move on another types of microbes. So let's talk about the bacteria. I mean the antibiotic. Antibiotic like the antimicrobial, that antibiotic also this substance which produced from the microorganism, which produced from some plants like vegetables and some animal tissue. It can be also synthetic, semi-synthetic, and the natural analogous. So the same thing like the antimicrobial. And the antibiotic is divided into two types according to the spectrum and according to the action. So it's divided into two types: a spectrum and action type of we can say just action. So if you talk about the spectrum, a spectrum is further divided into mainly two parts, but first is the narrow spectrum, and the second one is the broad spectrum, or wide spectrum, wide or broad, we can say wide or broad. In all also extended spectrum, but it's most probably in rare cases we use as extended spectrum, but discuss there also. According to the action is same like in a microbial. As we know that as I said, the microbicidal and microbistasis, they here also bactericidal, and the bacteriostatic. Means the bactericidal means the those antibiotic who belong which belong to the bactericidal, they killing the bacteria, and which antibiotic, those antibiotic which belong to bacteriostatic, that is the inhibiting the bacterial synthesis. So let's talk about now that this is the very understanding that we already understand the what is the bactericidal and what is the bacteriostatic. Now we'll discuss about the narrow spectrum and wide broad spectrum. What is this? Because a lot of drug is the belong to the narrow spectrum and wide spectrum. That is the very important to know. So let's talk about this one. First, let's talk about narrow spectrum. What is the narrow spectrum? Narrow spectrum means, by the word we can understand, that is the narrow. Means in this, whatever drug which belong to the narrow spectrum, they will just kill the particular bacteria. For example, if the any drug which kill that gram negative bacteria, for example, gram negative bacteria. So if any person to come, for example, if any patient to come my clinic, and I know that the person have the gram negative a specific bacteria, for example, gram negative bacteria is Escherichia coli, E. coli. Let's think about the Escherichia coli. So the persons came our clinic, and according to the patient's symptom, I take the sample and send to the laboratory investigation. And after the laboratory investigation, they send the report that the person has Escherichia coli. And I know that whatever drugs belong to narrow spectrum, their drug will be very sensitive to the Escherichia coli. So obviously on the time we can give, and the person will treat well, and they will not even create more side effect, because just narrow spectrum, they will just kill the Escherichia coli. It will not too much side effects. So this is the narrow spectrum. Means already we know that the person have this types of bacteria. On the time we have to give the narrow spectrum. The next one, wide spectrum or broad spectrum. What is the wide spectrum, broad spectrum? So wide spectrum or broad spectrum. In these conditions, for example, let's think about again that any patient come to our clinic, and the person's condition become more worse, and we don't know which types of bacteria is there, but we know, we confirm that the person have bacterial disease, not viral, not fungal, not any helminthes, and not protozoal. The patient have bacterial diseases, but which types of bacteria we don't know. Either it's gram negative, gram positive, which types of gram negative, which types of gram positive, we don't know. And for the further investigation, we have to take the patient's sample, and we have to send to the laboratory, I mean the for the identification of the particular bacteria. But it will take more time, and the patient condition has become too worse if we will do take the specimen, take the person any fluid or any cultures, any like the any sputum, to send the laboratory investigation. That's why on the time we have to use wide spectrum. Means we know that the person have bacterial diseases, but we don't know exact bacteria. So in these conditions we can give wide spectrum, because it can kill the gram negative, and it can kill the gram positive. And as we know that the most probably bacteria is the gram positive and gram negative. So obviously these types of whatever drugs belong to the wide spectrum, they will helpful for the person. But what is the bad thing? Bad thing, it can be start more side effect. It can be create the more side effect. That is the bad thing. But obviously in these conditions, first we have to save the person's life. Side effects can be automatically disappear in I mean the further. But first we have to we can say that treat the patient or save the life of the patient. So this is the wide spectrum. And one thing also here I have to mention, that empirical treatment or empirical therapy. What is the empirical therapy? Empirical therapy. Empirical therapy is the same thing as I said, that if any person or if any patient came to the hospital, came to the clinic, and the person conditions very critical, very serious conditions, means we don't have time to investigate which types of bacteria is there. We don't have time to take the person's sample to send the laboratory to further investigate of the bacteria. That time we have to use antibiotic in any cases to save the person's life. On that treatment, or that we can say uses of the bacteria, use of the antibiotic, that is called empirical therapy. Means without any investigation, without, just we know that the person have bacterial diseases, but we have to treat the antibiotic, any antibiotic that can be kill either gram any bacteria, or as we know that bacteria have most probably gram negative, gram positive. So in this case, most probably wide spectrum bacteria, wide spectrum antibiotic we use. That is the called empirical therapy. So these are the main importance about the antibiotic. And apart from narrow spectrum and wide spectrum, also we have extend spectrum. So extended spectrum. Actually, extended spectrum, this is the very special types of drug. So actually in this drug mainly used in the very typical bacteria which is not cured by the wide spectrum and narrow spectrum. Obviously narrow spectrum will not cure. On the time we can use extended spectrum, but the most probably we use wide spectrum because a lot of patients will come, and for the fast treatment always we use wide spectrum, because in wide spectrum even without any laboratory investigation, if we suspect the person have bacterial disease, so by the wide spectrum we can treat. In the extended spectrum also it's very rare, because if we first use the wide spectrum, obviously after using wide spectrum no any result will be developed. Obviously then you will try to investigate the particular bacteria, and after the if you diagnosis that this have the person have this typical bacteria, special bacteria, and that bacteria will not kill by the wide spectrum, so on the time we use extended spectrum. Which types of drug is belong to the extended spectrum we'll discuss. So first of all, this is the main basic criteria of the antimicrobial or antibiotic uses. So before the starting use the antibiotic or any antimicrobial therapy, this all criteria should be done. Then we should start the antimicrobial. So in the next part, we will first discuss about the all antibiotic drug, because a lot of drugs belong to the antibiotic. Then we'll individually drug will discuss one by one. So till then, take care. Thank you so much.